Okay, so we're going to get started with score functions. So from yesterday's lecture, does anybody remember the two fundamental challenges that we're able to address with PyRosetta type modeling? So we have the sampling challenge and the scoring challenge. Anybody want to brainstorm what actually is the challenge with, let's say, sampling? So we have to sample conformations because there are multiple conformations of amino acids together. Anything else that we're interested in sampling? Okay, so we might have to sample different sequence identities at different positions, and then maybe sample their conformations. So to go hand in hand with that, what are we scoring? Okay, so maybe we're trying to have some free energy calculations. Does that help our sampling process? Okay. What's the relationship between free energy and stability for proteins? So maybe we think that the lowest free energy is going to be the most stable. Okay. Can you think of a score that doesn't involve free energy? So maybe something that is probability-based and doesn't rely on free energy calculations or approximation. Interesting. So let's talk about how Rosetta will sort of score your conformations and changes to your sequence and how we might use that to address sampling problem. We're going to specifically talk about an energy function that was made in 2015. So this is referred to as Rev 2015. And this is a paper that will be shared with you. It sort of goes into detail in terms of different energy terms, and you can learn a lot by just staring at some of these tables. But we're going to talk about the actual concept and why we needed to come up with an energy function to begin with. Today and yesterday, you've taken a look at residue-specific energies. So with POS, you were able to calculate energies at a specific position. And when you were doing that and taking a look at sort of making perturbations at the phi and psi angles and looking at different POS energies, maybe you were able to see as you sort of got further away from the ideal coordinates, something about the energies changed. So it was sort of loosely hinted, let's just say, that setting different phi and psi angles is going to change the energetics for the small peptide that you were working with. And so we have to think about what in our, let's say, entire sort of Rosetta suite do we have to calculate some type of energy so that we can score different structures, different designs, and also sample perhaps multiple conformations, different sequences, and sort of guide our process towards successful protein design, whatever that means, right? And so yesterday you learned about Levinthal's paradox. So let's just talk about the paradox for right now. But essentially the idea is that if we were to sort of figure out every single possible configuration for every single residue and then the way they come together to make secondary structure and then tertiary structure, how long would it take us for the average length protein, which we said was 300? You know, the point of it was that it would just take incredibly long and that we would need a lot of time. And we don't have this, but we still want to sort of design proteins and make artificial molecules. There is a lot of impact in terms of, let's just say, protein-based therapies. And obviously, proteins bind drugs. Sometimes they are drugs. They're used in diagnostics. And so we don't have that much time, right? We want to solve these problems and do it right now. And so what we're going to think about is how you can sort of approach protein folding as a problem without doing this via brute force. So how do we fold proteins that we design. So how do you artificially design a protein and then take a look at either whether it would fold or what exactly it would fold into in its 3D conformation. And something that was mentioned earlier was that we can have some relationship between the most stable, or perhaps, let's just say, as we have our free energy minima, we're going to have sort of the most stable protein conformation. And so something to think about here is that as we go down the energy well shown here, we're able to have our protein sort of start folding by minimizing on whatever free energy that we're talking about. And in this case, something that I'd like to point out is sort of what we refer to as the native state at the very low energy conformation here. And so in general, just like the field of biology, in general, what do you think of when you hear the word native? So you have your native protein, maybe we'll say wild type. So just in general, what is that term used for? So some protein that naturally occurs, so maybe it's naturally folded conformation, we'll call the native state. And so something I'd like to, sort of a question I like to pose here is we're decreasing entropy and we're minimizing on free energy, and we're going from an unfolded structure to, let's say, something that's slightly misfolded, like a molten globule, into the native state. So once we make it here, do you think that that native state is the most stable our given protein conformation sort of can get to? Okay, so everybody agrees. Is it possible to have alternative conformations of a protein? Can a protein have two native states? Can it have three native states? Raise your hand if you think a protein, by this definition, could have three native states. So according to this definition of native state, where you're at the lowest energy conformation, can you think of there being multiple native states? So can you have two lowest energy conformations? So just to repeat that, actually just to draw that out really quick, perhaps we're thinking of something like that. Okay, so now we're perhaps thinking of something like that. Raise your hand if you think that the solid line is more likely. All right, so our explanation for why the solid line is more likely is that perhaps corresponding to each of these states, so for each, let's say, local minimum, perhaps we have some type of biological trigger. So that could be something functional. So binding a small molecule, for example, could trigger a different free energy response. And can anybody think of a way you could have the dashed lines where each of the local minimums are sort of at the same lowest energy? What we're hearing is that perhaps there was no clear advantage being in either state 1, 2, or 3, and that they were somewhat similar, at least, let's say, in evolution's eye, or something like that. Whatever this protein was evolved to do, it was equally able to in any one of the three free states, perhaps. Any other explanation for how you might have the dashed lines? Okay, so now we're talking about protein switches. So perhaps we have a conformational switch that's able to go between either 2 states or all 3 states, and let's say that perhaps it's equally likely. So that's a possibility. Yep. Once again, you could have it be the solid line if there is a trigger to the switching. Now, for the dashed line, the idea perhaps is that there isn't necessarily a single trigger that changes the energy landscape for the fold to switch. So what that means is sometimes in nature, you'll have simultaneous switches. So, sorry, it will be a spontaneous switch. So it will go between, let's say, 2 conformations very quickly, and then perhaps even an intermediate state, and it will just sort of cycle through. And so none of the individual states are, they don't require perhaps a signal anywhere in the environment, and it's able to change throughout rapidly. So we're going to think about protein rigidity in terms of dynamics. As well, we're going to sort of think about proteins breathing. So whenever we crystallize, we sort of look at the most ideal structure that we can have in crystal conditions, and then we have to sort of resolve the protein structure. But in nature, we know that proteins sort of have a little bit of flexibility, and they sort of move around. And yeah, we can look at a few conformations just showing in which regions you can have more flexibility. And that also relates partially to function, partially to just NNC terminals, to loops, etc. But yeah, I mean, this is really good insight. So the last question before we move on from this slide is, let's call this native protein, protein A. So protein A was, let's say that it's a rat protein, and let's say we're interested in studying it. So we took it from rats, and we did some experiments. We somehow figured out a way to get its full 3-dimensional structure, and we're calling that the native state. So we have its amino acid sequence, we have all the 3-dimensional information, and then what we do, of course, since we're working on PyRosetta, we put this into our little notebook and then take a look at whether we can make some random mutations. And let's say that we have a way of searching for which mutation would possibly be beneficial. Let's say that we have a very large database of other rat proteins that we can sort of look up against. And then using Rosetta, we figure out a way to change some of the positions and make them slightly different so that they still look like a rat protein, but it's different than the native state that we sort of have the structure of. What do you expect to happen then? So let's say we take this, we make some perturbations. Let's include some phi and psi perturbations, so that you already know. Let's also say 3-point mutations. And then, does it still have the same energy as the native state? No? Okay. It definitely depends on what you changed. So, okay, let's pose another question. Is it possible for it to have lower energy than what you started with? Is it possible? Is it possible to go beyond this local minimum? No. Okay, so now we're thinking, well, maybe it is possible to search and find and make some changes that allow you to access even a lower energy conformation. Okay, so then one final question about this is, well, if that's true, is it correct to call this the native state? If I could sort of search and find a lower minimum, is it what's the right way to figure out what the native state is? Because it's not really about it being wild type anymore, right? It's not that it comes from nature. If the native state is going to be whichever is the most stable in terms of free energy minimization, then I can go beyond what nature has in store for us. So just keep that in mind. And with all of this, we can sort of start discussing exactly why we need a score function. As a few of you pointed out, in terms of what perturbations or mutations we make to our protein, we don't actually know if the energy is going to go up or down. So we need a way to keep track of all of this and make the right changes. And so we have to score these changes and perhaps only consider the ones that are favorable in terms of some energy or some approximation of free energy. And so in Rosetta, in order to have a way of sort of keeping score of changes that we make to protein structure and sequence, we're going to talk about how we might want to represent a pose and sort of make changes either in this full atom representation here or a centroid representation here. So just by looking at these two images, what's one observation between having full atom versus centroid? So we have some atoms that got grouped into sphere right here compared to the full atom, which is showing every single atom as per the name. We're hearing that the centroid is less complex. Perhaps it's coarsened. Anything else? Yeah. Okay, so perhaps the side chains are grouped together to make a centroid. So here's a question. Now that we have some differentiation between having every single atom represented, or possibly in some cases every single heavy atom represented, you learned about that yesterday. What's an advantage of having this centroid representation? So we have a way to analyze and screen for designs a lot quicker because it's coarsened and we're calculating for less atomic energy. Instead, we have centroid energy. So that will come in handy. And let's say that eventually you have a sort of population of designs that make it through your centroid-based scoring. Would you still want to analyze what their full atom energy pseudo-landscape still looks like? Maybe after they make it through the cutoff, later in your pipeline, you sort of go into more details and analyze what happens in the full atom scheme. So that sounds reasonable. And in terms of what energy landscapes look like, we can take a look at centroid energy landscape and compare it to the full atom energy landscape. And obviously this is in 3D, but try to somewhat relate it to the other 2D energy landscapes that we've looked at throughout today and yesterday, possibly. So, I mean, these two pictures look different, right? What's the difference? The explanation that we have is they tell you the same story, but the full atom energy landscape is giving you all the details. Whereas the ideal centroid energy landscape is giving you sort of the gist of it, but perhaps it's quicker, the search is smoother. And so in general, for native proteins, if we were to actually take a look at what the energy landscape looks like, and we had a way of determining this, either through some simulation or some experimentation, we would see that the energy landscape is rugged. So it's going to look sort of a lot rougher than the ideal centroid energy landscape that we have. But obviously, it is easier to search through a smoother surface and sort of figure out how you can minimize on that, as opposed to having all of these sort of peak at different spots. So this is like a little bit noisier. So we can smooth on that noise and still reach our minima that we're interested in finding. So this sort of allows you to search more efficiently, as we pointed out earlier. And now let's talk about Ref 2015 score function. So one thing to consider is we only talk about this score function in detail, but there are different score functions in PyRosetta. So there are multiple versions of this in terms of the way it was developed, as well as sort of trying to figure out what energy function works at all. And besides that, some of you might know about perhaps membrane proteins, and for insoluble proteins, the community sort of works with different energy function because the overall energy landscape looks different. And anybody know the difference between membrane proteins versus soluble proteins? So maybe it's connected to the bilayer somehow. Do you think that it always has to be exposed on the surface? Can it be fully buried into a membrane? Maybe, right? So what's an energy term that's relevant to membrane proteins? What do we know about the bilayers? Right, so we know that there has to be a very heavy hydrophobic term so that we can calculate it appropriately. And so the environment of the protein influences the overall energy, and then we can make our calculations more accurate by accounting for that. So let's go back to our sort of general idea and our general score function that we can use and talk about Rev 15. So there is going to be multiple interactions and different types of energies that we consider for each residue position. And yesterday you had sort of a shorter discussion on what energies in general sort of drive molecular interactions in nature, or more so in physics. And before we get into this, do you remember from yesterday's discussion what types of energies we have? So we have electrostatic interactions. We talked about the hydrophobic effect just now. Anything other than those two? Let's think about perhaps the sidechain group for a little bit. So what was the difference between, let's say, glycine and arginine that we talked about yesterday? So we know with glycine, sort of the sidechain atoms don't occupy as much space, nearly as much space as all of arginine's sidechain atoms. So for a bulky residue like arginine, do you think we would have a problem trying to fit all of the sidechain groups into some space? Let's say that you have a 3D structure. Let's say it looks like something like that. And let's say that this is entirely made of bulky residues. So every single residue here is extremely bulky, and it has sort of very long side chains that are trying to come and fit together. Is that energetically favorable? Why not? What happens when atoms get too close to each other? They repulse? They push each other away? Okay, and do you remember the name of the potential that sort of describes that repulsive behavior? Yeah. Okay, so we're hearing Lennard-Jones potential, and besides Lennard-Jones, we can also think of just the fact that you can't have two three-dimensional objects occupy the same exact volume. So we're hearing excluded volume. Also, besides that, we might want to think about sort of how much space there is in general for everything to sort of come together and fit nicely, making the entire structure possible or feasible for folding. And so to tie it back into different types of interactions, we're going to have so everything that we talked about, and we're trying to combine these to make some approximation on the energy total So every energy term is going to have its own weight, and we're trying to put together interactions on the basis of perhaps hydrogen and disulfide bonds. So which amino acid makes disulfide bonds? Okay, and which amino acid makes hydrogen bonds? Okay, so slightly tricky question there. Is it possible to make hydrogen bonds with the backbone atoms instead of the side chain? Right. So you're going to have sort of residues that can make hydrogen bonds and fit into a helix. And so all of those secondary structures are held together with the help of hydrogen bonds. So you definitely want to keep track of that. Also, we're going to have non-bonded pairs energy. So this is going to be sort of based off of how far apart you have your, let's say, two residues. And then if you're running this in centroid configuration, it's going to depend on how far apart sort of the average center of mass is, representing the rest of the protein coarsely. So we talked about torsions, plus you did a lab on this. What are torsions? What angles for backbone torsions? Okay, so we're going to have some energy associated with what type of torsion angles or combinations of torsions can sort of be accommodated by a backbone. And so in your Ramachandran plot, you can sort of figure out which sort of region is impossible to occupy if you wanted to have a regular, or let's just say if you wanted to have an idealized secondary structure conformation. And other sort of environmental or just in general in our molecular assembly, there are other factors that might influence the energy landscape. Can you think of anything just in general? What changes the energy? So we were talking about the differences between these two energy landscapes. And if we try to relate this back to everything we see here, what's an example of an environmental factor that changes the energy? Okay, pH. What is pH? Okay, so maybe something about concentration of hydrogen. Anybody else? Yeah. Okay, so we're going to have temperature. That's going to change our energy. Makes sense. Anybody else? Yep. Okay, so maybe the solvent hydrophobicity influences things. Anybody else? So perhaps for the ligands that are recognized, their concentration could change things, or whether they are sort of fully absent could change things for us. Anything else that you can think of? All right, well, this is plenty. So with all these considerations, how do we ever come up with a very generalized scheme and have all the protein structures that, let's say, we have available, sort of have them fit into our idea of this new energy function and to be able to score them accurately? And so it already sounds like a very difficult problem. So you're putting together sort of all the degrees of freedom that you can have and have a way of evaluating exactly what happens given different chemical propensities and different types of conditions. And there is a lot that goes into sort of figuring out exactly how to weigh which term sort of higher or lower and for which condition, and exactly how everything could come together. So we don't go into the details here, but you might want to think about how far away two atoms have to be to sort of start either repulsing or attracting. And in general, you can take a look at how that changes perhaps in combination with other types of energy terms. So we briefly talked about solvation in terms of, you can think membrane proteins. And with hydrogen bonds, so you're going to have a chance to really analyze what happens to hydrogen bonding in terms of its distance and, you know, sort of what distance range is feasible. Anybody know hydrogen bond distance? If we could get a quick maximum minimum on that, okay, we're hearing 4 angstrom. Anything else? Okay, so on the graph there is 2 angstroms. Okay, so let's just say we have an upper lower bound, and it could definitely vary based on sort of which atom and exactly the surrounding, but it's going to be possible to have, well, we don't talk about this in detail, but you can sort of have, let's say, 2.3 angstroms to 3.5, with slight deviation from that is going to be possible. And for disulfide bonds, we also are able to calculate energy because we can study these atomic sort of energies in experiments as well. So you can break a disulfide bond and just see what happens and figure out a way to relate that and use it in your score function. And we also talked about torsion, and we know for a helices sort of ideal coordinates what the torsion sort of energy should be, or exactly how to idealize those coordinates so that it minimizes on the overall energy term. And then the sidechain conformations. So something we didn't talk too much about is sort of the conformation space for rotamers. So just like you have dihedral angles in the backbone making your torsions, you have dihedral angles making up the sidechain conformation. So if you think about the plane that I showed you moving for phi and psi yesterday, we're going to have sidechain angle, we're going to have sidechain dihedrals referred to as chi1, chi2, chi3, and then you can have up to chi5. Which amino acid has up to chi5? There's only one. Which residue has the most sidechain atoms? Louder? Yeah, so arginine is going to have up to chi5. And so if we calculate all of those dihedrals based on, let's say, the surrounding context as well as the backbone conformation, we can figure out if they're rotameric and if they're in the ideal sort of coordinates for where the sidechain heavy atoms should be. And that's going to have a lot to do with energy in terms of both the sidechain, and you can think sterics, you can think hydrogen bonding. It's just going to influence all of that. And then we have some sort of residues that are more special in how their sidechain interacts with the rest of the protein. So from this, the most intuitive example is going to be proline. What is unique about proline? So proline sidechain essentially makes a ring with the backbone. And so the proline sort of sidechain is different in that it can't be treated independently from the backbone as you would with the other residues. And so it also changes sort of what we can design for certain secondary structure regions. So we didn't explicitly do this, but it might be important to think about sort of when you look at your helices and sheets, which residue identities do you never see in those secondary structure units. So maybe for loops, it's possible to see just about any residue go in a loop and still sort of keep the loop conformation. But you can make a point mutation so damaging or so disadvantageous to your secondary structure being, let's say, helix, that it actually breaks the entire region. And so do you think that a proline could sort of break secondary structure for a helix? Maybe with what it does in that ring, it could possibly be breaking regular and organized hydrogen bonding patterns that you want. So in general, it's good to think about what residue identities sort of are known to cause problems in some secondary structure or the other. And then we mentioned there being, so some of you pointed out things listed here already, but we also talked about membrane score function. So scoring in the membrane is different, or scoring membrane proteins is different than scoring soluble proteins. We can have other types of biomolecular interactions. So the assembly could include some DNA or RNA, and so that changes the energies. So the question is, why do you think we care to represent or sort of change our energies based on DNA or RNA? Why can't they be treated the same way proteins are treated as far as we're concerned with energy? So their structure is different. What else? Or what about the structure is actually different? Are they truly smaller than proteins? You're saying no, why not? So you can have a very long DNA sequence. Are they structured like proteins? You're saying no, why not? So we're getting that they're helices. Are they always helices? Okay, so they have their own distinct, both secondary and tertiary structures. So you can have actually very defined regions in RNA, and well, sometimes it's possible to have sort of shapes that are possible to accommodate by RNA, as you would see in proteins. So sometimes you could have, let's say, you have your protein bound to a protein. That's also maybe possible to recognize some type of RNA. You could have them sort of making up the same shape, but that 3D structure is made from different sort of monomers. So their residues are entirely different. And so the flexibility is going to be different. And so the secondary structure units that come together to make the same shape at the interface to recognize that protein is going to be different. And so you're going to have different, at the very least, flexibility. So that, at least to me, is the most intuitive one, is it's just going to look different in terms of how much you can sort of, let's just say, collapse it, versus how rigid it is. So you can't really sort of compress and make the structure really small, etc. But also from how flexible it is, it's possible for RNA to take up so many different shapes. And it does in nature for various functions. And then what we didn't cover is, so what I'm describing was actually molecular docking. So whenever you make an interface and you sort of have a binding target and you try to sort of see how they come together and dock in 3D space, that itself is a very difficult problem. And having docking sort of simulations, you're going to require different types of energy. So if we're concerned with docking, on top of our normal score function, what additional terms we might consider? So what additional terms might we consider given that we care so much about making an interface? One idea is for every position that we know to be at the interface, we might make it more sensitive, so we can dial up energies at the interface, or sort of score heavily at the interface so that we get what we want. But is it always true that in docking challenges, we know exactly where the interface is? So in nature, let's say I give you a pathogen and I say, for this pathogen, sort of come up with a way to bind, just bind to it, making a brand new protein. Would you know where to start in terms of where it should be docked? Would you want to try multiple interfaces, trying to make different interfaces with the pathogen? So in that case, perhaps you need a way to score different types of sort of docked overall complexes you have and see if they score well in terms of the interface. And so that seems perfectly reasonable and something that will be useful for real biological challenges that we're all interested in. And then, yeah, there are many more examples. Proteins also sort of make symmetric assemblies, so they come together to make a symmetric complex sometimes. Anybody know maybe an example of this where you have protein symmetry? Yeah. So antibodies do have the heavy and light chain, and they come together, but they're actually not symmetric units, so they're not copies of the same chain. Oh, yes. The entire antibody is symmetrical, sort of in terms of its. That's true. Yeah. So with hemoglobin, great example. So with that, you also have sort of binding pockets that are repeated, and so you have different subunits come together, but it's also symmetrical. That's right. And there's many other examples. Actually, you'll be designing some symmetric proteins very soon. Not today, but pretty soon. And so hopefully that's of interest to everybody. So one thing to mention is the energy units for Ref 15 are going to be arbitrary. And so they don't sort of relate, or they don't have conversion to real energy units. The idea is to get some type of ranking in these arbitrary Rosetta energy units, but you're not necessarily trying to find exactly how it would convert to real energy. So the idea is to have a way to search, but it's not a one-to-one sort of perfect method of scoring your designs. So we're calculating scores per residue, and you can sort of average every single residue energy, or you can sum them up, or you can consider only the interface residues energy combined, etc. So different types of different ways to tailor the score. And you also sort of need to score in, I guess, various conditions depending on your problem. So if it is not entirely, if your problem is not entirely well addressed, it's also possible that no energy function in Rosetta really gives you any insight. And at that point, you might need to make adjustments by yourself and write it a little bit for just your problem. And so it's also possible that for some native proteins, it doesn't really correlate well either to the native state or to some of the known mutations or to any other stable design that you come up with. And so just keep in mind about how reliable the score might be.